Alright, now let's talk about the nucleotides that make up DNA. There are two types of nucleotides, the purines, which are adenine and guanine, and the pyrimidines, which are cytosine, thymine, and uracil. Uracil is only found in RNA, and thymine is only found in DNA. You can remember these using the mnemonic pure as gold, since the purines are adenine and guanine, and cut the pi, since cytosine, uracil, and thymine are the pyrimidines. Alright, let's see if you're paying attention. Adenine, is it a purine or a pyrimidine? Right, adenine's a purine. Remember, pure as gold. So fortunately, you don't have to memorize the structures of these for your exam, but there are some features you should be familiar with. First of all, the purines have a double ring structure, whereas the pyrimidines have only a single ring. You can use the cut the pi mnemonic to remember this, because the single ring looks kind of like a pie. The only other things you have to know for the step 1 are that thymine has a methyl group here, and cytosine can be deaminated to make uracil. So why do we care about all these functional groups anyway? Well, the differences in the locations of the ketones and amine groups determine where and how many hydrogen bonds are made when nucleotides pair up in a double helix. Guanine and cytosine pair up by forming 3 hydrogen bonds, shown by the dashed lines here, whereas adenine and thymine form 2 hydrogen bonds, 1 and 2. So which type of pairing is stronger? That would be the GC bonds, which means it requires more energy to separate DNA strands that are rich in Gs and Cs, compared to those that are rich in As and Ts. This ties into the concept of the melting temperature of DNA, which is the temperature you have to reach to separate two strands. DNA is double-stranded at lower temperatures, but as you heat it up, you can get the strands to separate. So would G and C-rich DNA have a higher or lower melting temperature than A and T-rich DNA? G and C-rich DNA would have a higher melting temperature, because again, you need more energy to separate these strands, since they hold on to each other more tightly than the AT bonds. Can you think of other properties of DNA that might affect its melting temperature? One of the other most important ones is DNA length, since shorter stretches of DNA are easier to melt apart. This kind of makes sense, right, since there are fewer total bonds. So who cares how strong DNA bonds are anyway? Well, we'll come back to this in the lab techniques section, since in some types of experiments or diagnostic tests, like PCR, the melting temperature of DNA is very important. And it's also important anytime you have to transcribe DNA into RNA, or replicate DNA, because the proteins that do these things have to first find AT-rich regions so they can separate the strands. Alright, to drive this home, let's do an example. Which of these pieces of double-stranded DNA has a higher melting temperature? I'll give you a couple seconds to think about it. So this would be the bottom strand, since it's both longer and it has more G's and C's, both of which help the strands stay together and increase the melting temperature. Alright, shifting gears a bit, you should also know what components are used to build nucleotides. I'll discuss this more in another slide when we go through the nucleotide synthesis in more detail, but for now, you should know that they're made primarily from amino acids, with purines requiring aspartate, glycine, and glutamine, and pyrimidines requiring aspartate and carbamoyl phosphate. It's convenient that the first letters of the amino acids used to make purines are A, G, and G, since again the purines are adenine and guanine. Alright, the last thing I want to cover here is nomenclature. We've already covered the names of the bases, but nucleotides are more than just bases. They also need to have a sugar and a phosphate. What kind of sugar is it? Well, in RNA it's ribose, and in DNA it's deoxyribose. And what's the difference between these two? In this diagram, you can see that the 2' carbon in the sugar can have either a hydroxyl group, which makes it ribose, or just a hydrogen, which makes it deoxyribose. So when we say a nucleotide, we're talking about the base, the sugar, and one phosphate. If you just have a base and a sugar, that's called a nucleoside. This also affects the names of specific bases. For example, the nucleoside version of adenine is adenosine, and the nucleotide version is adenosine monophosphate. If you add 2 more phosphates to adenosine monophosphate, you get adenosine triphosphate, which as you probably know is the main energy currency of the cell.